Hello, my name is Sofia Jacobson, and today I'm going to be talking about my research poster. My research is trying to understand different additive methods to create self-assembled monolayers. This field is commonly known to research and dwell into how to reduce the amount of defects that are formed on these layers with self-assembly, which is in nature done, but trying to do that in industry is difficult. So we have to utilize different procedures in order to create these good defect-free monolayers. So within my project, I'm trying to explore if reducing the amount of deposition time of organosilane solution monolayers can be done, because in the past, previously in our background, we've done 24-hour OTS depositions and then over time decrease it to 1-hour depositions. And within my research, I'm trying to see if 5-minute depositions is sufficient enough to reach that full thickness of monolayer. The full thickness is 26 angstroms, and by doing 5 minutes of depositions, usually that doesn't reach that thickness, and so I'm introducing cycles of cleaning steps. So this is going to include an oxidizing base clean as well as a chloroform clean on my sample, and then doing cycles of that and the 5-minute depositions in hopes of reaching that 26 angstroms that we're looking for. So with this, we have introduced the cleaning cycles and the OTS deposition 5-minute cycles, and by doing them within these cycles, we hope that with each cycle we can get closer to the thickness as well as reducing the amount of defects on the layer. So looking at our results are currently inconclusive because we don't have enough data points to make an association with that yet, but based on what I have right now, we can see that with each cycle, the error bars actually get smaller. And this is important because when the error bars get smaller, this means that the data is closely related together. And this means that because of that, we can kind of infer the uniformity of our layer, and then it's actually pretty uniform, meaning there's less defects. And these can also be inferred based on our water contact angle, which supports the thickness data. And our water contact angle just looks at the hydrophobic surface of the OTS, because the OTS has a methyl group on the end. And with this, if the data is pretty uniform and the layer is pretty uniform, we would get a contact angle of around 110. And if our uniformity was not nice, then we would get a lower contact angle. So with this, we've inferred that with each cycle, we reach that desired thickness, lower the error bar values, and in addition, it is heavily supported with the water contact angle measurements. So overall, this data is inconclusive, but we are planning on continuing this data to develop more concrete answers and trying to finalize this procedure in hopes of other industry and research labs utilizing it. Thank you very much for listening, and yeah.